Here's a quick tutorial to show you how to plot your data for Lab 6 in Microsoft Excel. If you choose to plot your data on graph paper instead, many of the principles are the same. Hopefully you find this tutorial useful. Here's a table of some sample data. Your data, of course, will be different, but some of the principles will be the same. You can see water is 0, 0, 0 all the way across, which is good. It shows there's no fermentation there. The corn syrup, on the other hand, is highly active, and the numbers are quite large when you get to the 45-minute part. Everything else should be in between. You can see there's some sample data there for stem extract, kernel extract, cob extract, and amylase, and so on. And so how to make sense of these numbers is we're going to make a graph, and I'm going to show you what you need to graph and how to do that here in this tutorial. For our graphs, we're going to put the volume of carbon dioxide produced on the y-axis and time on the x-axis up to 45 minutes. We can throw in our water values. It's all going to be 0, 0, 0 all the way across. And what we can do is connect the dots. In this case here, we don't necessarily have to look for a line of best fit because it's all flat, pretty easy one to do. We'll put another one on the graph here, and this one is not going to be flat. This doesn't represent our data necessarily. This is from a different semester, but you'll get the idea of the shape of most of the other curves. So there's another plot. Most of the other curves will start off relatively slow. They may not produce carbon dioxide in the first 5, 10, or in some cases even 15 minutes, but then eventually they start producing carbon dioxide and you get into an exponential phase. Eventually, at some point, maybe around the 30 or 40-minute mark, it starts slowing down and it plateaus at the top. So we have a little bit of a sigmoidal or an S-shaped curve. Like before, you can connect the dots or you can find a curved line of best fit. It doesn't really matter, but do draw lines so that you can see the different data points and the different lines really well. So what you're going to do is put all 6 of your plots on one graph. And I have a small Excel tutorial to show you how to do this in a moment, and hopefully that will help you to produce your graph. The next step in our data analysis is to calculate the slope of each of our graphs. So what I want you to do is calculate the slope of the linear exponential portion of the curve. So what does this mean? It means we're going to ignore the flat part at the beginning and the flat part at the end and just get that exponential part in the middle. So in this particular graph, what I'm going to do is select that middle portion, and I'm going to select the time points at 15 and 30 minutes. I'm going to calculate my slope for that particular range there, and the slope is equal to rise over run. So in this case here, the rise is 0.83 minus 0.18 milliliters, and the run is 30 minutes minus 15 minutes. So my final value for my slope is 0.043 mils per minute. So this means that for this particular treatment, we're making 0.043 milliliters of carbon dioxide per minute. So you're going to do this for all 6 of your plots, and what I want you to do is put all of them on one graph.
You can see here I have the data. It's all inputted into the spreadsheet and ready to go. So what I'm going to do is first select the data, so B3, and highlight all the way to L9. And it's pretty simple. We go up to Insert. I'm going to go over to the chart area. You can see there's lots of options. I'm going to select this one, which is the scatter chart. I'm going to select one of these options. You can see there's lots of options there. I like the one where it connects the dots. And there's my chart. So you can see it is looking pretty good and it's almost ready to go. It's not quite fitting all of the Biology 107 standards. There's a few things we need to do. First thing we need to do is add axes titles. So what you need to do is click on your chart and then go up to the top and click on Design. So if you go over to the far left now, Add Chart Element, we can add axes titles, which are right here. I'll start off with my horizontal title. So my x-axis is time, and so the units are minutes. And I can add my y-axis title. So the title here is going to be volume of carbon dioxide produced, and the units are milliliters. I'm going to do one more thing, the little 2, I'm going to make it a subscript. I go to Home, and I select Font, and find the subscript option to make it a little 2. Next thing I'm going to do, I don't really like my legend being on the bottom. I kind of like my legend on the right. So I'm going to right-click that, Format Legend, and you can see there are some options there, Legend Position. I'm going to click Right. My second note says use a different symbol for each line. So if you look at your graph, you may notice that all of these lines have the same symbol. They're all little circles. So that's a problem, because if we're going to print this off in black and white, we won't be able to tell which is which. It's all going to be grayscale. If I'm colorblind, or the person looking at your graph is colorblind, they won't be able to tell the difference between red and green, or even sometimes the different hues of blue are a little bit hard to distinguish. So what you want to do to change your symbols is click on the symbol like this, and you can right click and go down to Format Data Series. It should pop up here on the right. We're going to click on that little fill bucket, and we want to go here to marker. If you want to change the color, you can do that. I don't think that's necessary. These colors aren't too bad. And I'm going to go with marker options, built in, and what I'm going to do is select a different symbol. So I will select the square for this one. I'm going to make it a little bit bigger too, so it's easier to see. And there we go. So I'm going to go ahead and change the symbols for all of the lines, and then I'll see what note number 3 says. There's the final product. My graph has those nice little symbols, a different one for each, and a different color, so it's much easier to interpret. My third note is saying, hey, let's put the title below. So Excel has this default where they put the chart title up at the top. So what I'm going to do is just get rid of that. And what you can do is just insert the title either in Excel, or you can add it in your Word document and just type it in below. So this is Lab 6. So this will be figure 6-1. And I'm going to write myself a descriptive title. And my title is, of course, going to have the binomial name of the organism. In this case here, the organism is yeast. The binomial name you can look up on the internet, or you can look in the lab manual. It's found in both places. Now what I'm going to do is show you how to calculate the slope of the line. It's really important that you calculate the slope of the exponential portions of the lines. So for calculating your slopes of your lines, it can be easily done in Excel. We'll do the water for starters. The water one should be relatively simple because, of course, it's just 0, 0, 0, 0, and so on. And of course, the slope is 0, but I'll show you how to do that anyway. One thing that you can do is make a copy of your Excel file and do this for each of your slopes, or you can do it all on one graph if you like. It's just going to get really cluttery if you do it all in one graph. So what I'm going to do is actually delete all the data for everything except for water. You can see there's my water curve there at 0. If I click on my curve, if I right click and go to add trend line. So if you go down, scroll down, and at the bottom it says display the equation on the chart. And that's what's done right here. You can see my equation here is y equals 0. I'll make that a little bit bigger so that everyone can see it. And that's the equation on my water line. So now let's try looking at one of the other curves, maybe the corn syrup. Here's my curve for the corn syrup. You can see I still have the water on that curve, but I've deleted all the other data so that I can see the curve really easily. Like I said, it's much easier if I do this for each separate set of data points. A lot less cluttery, a lot easier to see, a lot easier to manage. So as I mentioned, you want to get the data for the exponential portion of the curve. So we want to miss this, we don't want this beginning region where it's kind of flat. We don't want this end region where it's starting to slope off. We want this region here in the middle on the curve. So the easiest thing to do is actually just delete that data. So here we go. I'm going to delete the data from this curve, make a copy so you don't have this deleted permanently, and I'm going to delete those last 3 points as well. So there we go. Now I have the exponential portion of the line. All I need to do now is find the trend line. So just like before, add the trend line, scroll down display the equation on the chart. And there's my equation right there. So I'm getting the slope and everything. I'll just make that a little bit bigger so that you can see that. There we go. And so for the corn syrup, this is the slope that I'm getting. So now after this, you want to do this with a stem extract, kernel extract, cob extract, and cob extract in amylase, and input the slope values into table 6-3.
Once you've produced all your graphs and you've figured out all the slopes of the linear exponential parts, you're going to input this data into table 6-3, and this here is your final data. So when you're discussing your hypothesis, when you're discussing your conclusions, this is the final data that you're going to talk about. You're not going to talk about how much carbon dioxide has been produced. You're going to talk about the rate of carbon dioxide production. This is the important essential data for this particular experiment.